Okay. Dr. Gary Aston Jones, welcome to the podcast.
Thank you, Mark. It's nice to be here.
Thank you. So, so you are a brain expert, right? A brain expert at the Rutgers Brain Health Institute. Tell me a little bit about yourself and, your expertise and, and how in the brain and, what kind of work you're doing.
Sure. Yeah, I got my PhD in neuroscience at Caltech and have studied systems in the brain for over 30 years now that are important in addiction, cognitive processes, and especially in motivation. Very interested in mechanisms that underlie motivated behavior, especially motivated behavior that is reward driven.
Okay. And now you said Caltech. That's where rocket scientists go to study. So are you?
Yeah, they have that's where JPL is, and right, they have a lot of very strong programs. I was in the biology program there. They have a strong biology program as well. So yeah, I was very fortunate to be there. That's where I really wanted to go, and couldn't believe it when I was able to. And it was a magical time. Richard Feynman was there when I was there, and we'd see him hanging around, and be able to talk to him and other people. John Wheeler, would come through. All these just luminary folks. So it was in a very exciting time.
Yeah. Yeah, that's incredible. Yeah, I love watching those excuse me. Yeah, those videos of the that most recent Mars rover landing, where the thing drops
Right.
itself down on the surface and has to find its own landing place and everything. Those are really fun to watch.
Yeah, 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 they are. I had friends that were in the planetary sciences department there. One friend, Carolyn Porco, is really well known now. She led the Cassini imaging project to image Saturn. She and I have been, in communication and reliving old times. And we used to go rafting down the Colorado River and the Middle Fork of the Salmon River and just have a lot of fun together as graduate students. It was such like I said, an exciting time.
Oh, did you ever come in contact with Carl Sagan, the famous astronomer?
I did not, but Carolyn won the Carl Sagan Award.
Oh, wow.
So, but no, I never met Carl.
Oh, okay. So yeah, let's get back to the brain and the topic that uh, we're supposed to start with here. You were doing some research into some kind of a sleeping medication
Yeah,
to help with addiction, and normally we try to avoid sleeping medications. When people ask for a sleeping pill, I'm like, oh no, those are bad. The Ambien and all the
right,
benzodiazepines, they're uh, somehow neurotoxic. Especially the long-term use. And
yeah,
I'm sure you know a lot more about that than I do, but I just know that they're not good for maybe temporary, but even temporary short-term use. We don't want people to get too used to those things, but that's
right.
not. You're working with some other kind of sleeping medication.
Yeah, I am. I'm working with some newly developed sleeping medications that work in an entirely different way from what's called the Z drugs, like Ambien, Zolpidem. Those drugs and benzodiazepines, the concern is they can be habit forming. Um, and these newer drugs are based on a system in the brain that uses a neurotransmitter chemical called orexin, OREXIN. And that was a system discovered in 98, and then found in 99 to be very important in narcolepsy and in driving waking from sleep. Drug companies immediately started developing compounds to block signaling in this system with the idea that that would then promote sleep and help treat insomnia by a very different mechanism from the Z drugs or benzodiazepines. Um, and in fact, they do that. At the same time, we were doing studies in addiction, and we noticed that cells that make this orexin signaling transmitter were activated in situations where animals were looking for drugs. And that stimulated a lot of experiments by us and others that now consistently show that this orexin system is also very important in driving motivated responding for salient rewards, especially for drugs of abuse, very broadly across all classes of drugs of abuse. So cocaine, opioids, alcohol, nicotine, everything that's been examined so far engages the system. And if we are able to block signaling in this system, we can block the hypermotivation that occurs in our animal experiments for different drugs of abuse. And there are recently results that indicate similar findings for human addiction patients as well.
Okay. And I hadn't really heard much about this, really nothing about this, to tell you the truth.
Yeah.
How is it that, is this something that do you think that this might be like a really significant effect that um, it might make the difference between someone going out to use more methamphetamine or cocaine or heroin or fentanyl? And if so, like why is, you would think this would be headline splashed everywhere, like that everybody would be getting real excited that they can use this drug already out there. Maybe doctors could prescribe it off label or whatever.
Right. Doctors could prescribe it off label, and I think they will. I think there will be also similar drugs developed for addiction. There are three drugs available right now that block signaling in orexin pathways from three different companies, Belsomra, Quivic, and Dayvigo. They all are blockers of orexin signaling. The one, Belsomra, has been studied, I think, the most in human patients and in animal subjects as well. And even in humans, it's shown to facilitate abstinence in a taper program where addicts will take less buprenorphine and be stable with less or no buprenorphine as long as they're taking this Belsomra. So it's consistent with a litany of animal experiments showing that if you block signaling in orexin, you can decrease this hypermotivation that is associated with stimuli that were previously associated with drugs of abuse and that drive a lot of the craving and drug seeking that occurs and lead to relapse in addiction subjects.
Yeah, and I'm actually looking up. I started looking up Balsamra,
Yeah,
and it says it is a control drug, Schedule 4.
it
Um,
is.
what does it have some abuse potential, or is there any concern about that?
The FDA had concern about that. I think it's misplaced. I think the reason they scheduled that and the other two as well, all of these drugs are Schedule 4 in the FDA, is because they are sleep-inducing drugs. And so the FDA decided to schedule them just like they schedule zolpidem and benzodiazepines, which is Schedule 4. And that was in the lack of evidence that it was a problem. I think it's just being conservative on the
Yeah.
part of the FDA. The companies certainly argued against that schedule, but we're not successful. Our evidence indicates it's not necessary, but yes, they are Schedule 4.
Okay. Yeah, and even the benzodiazepines apparently have, don't have like a really high addictive potential. People tend not to even really get addicted. They become dependent on them.
Right. That's right. They become dependent. They become kind of habitually dependent. Also, if they stop abruptly, then there are repercussions. When they suddenly terminate benzodiazepine administration, we don't see that kind of thing with these orexin antagonist drug. We don't see a sort of rebound, for example, in any of our animal studies.
Okay. And now you've mentioned a person who might be opioid dependent, needing less buprenorphine or Suboxone. Um, does it in any way possibly help with the physical withdrawal syndrome that happens as a person takes less and less buprenorphine?
Yeah, there's also evidence that it works for that. An early paper, blanking on the author's name right now, Ralph DeLeone, found that orexin antagonists are effective in decreasing withdrawal syndromes, again, in animal studies with opioids. That might be part of how they're able to facilitate abstinence, but we see very marked effects in decreasing what we call demand for drugs. We use a behavioral economics paradigm that quantifies the amount of demand or motivation for drugs. And in the absence of withdrawal, we see very clear decreases in demand and motivation for drugs. So it might be that it acts in both ways, like decreases of motivation independent of withdrawal, but also attenuate withdrawal signs.
Okay. Yeah, that would be a major benefit to, because there's not really a good exit plan for buprenorphine or Suboxone.
Absolutely right. No, those are opioid substitutions, right? That you're basically you're taking an opioid to treat opioid addiction, and currently there is no non-opioid therapeutic that's effective for opioid use, and this would be the first real example of a mechanism completely aside from opioids that would attenuate opioid use. I think part of the reason that it's not splashed in the headlines is because the companies are marketing these drugs to treat insomnia. That was the first finding, and that's a bigger market, I think. And so they want to market them in that way. A lot of times companies don't want to confuse their patient population with another indication, especially an indication like addiction, which has some negative connotations.
Yeah.
I think the way it'll move forward is the companies will develop other drugs that are very similar, probably, to the ones they now have for insomnia, but they'll market them for addiction separately. And they will turn out to be quite useful. It will require more clinical trials than have
Yeah.
occurred so far. The clinical trials are very early days, so it's going to take a few years of more clinical trials. But my prediction is these compounds will be found to be very useful for hypermotivation associated with abuse of a wide variety of drugs, and even for eating disorders. I think it will turn out to be useful
Yeah.
because there's a lot of shared circuitry between eating disorders and drug abuse.
Yeah, I was just about to ask that. You actually jumped right in my next question. A
Okay.
huge addiction problem, food addiction, or maybe it's carbohydrate addiction, binge eating, and there's not really anything good for that. I think they're using some diabetic drug for that. Naltrexone was combined with something else, and I haven't heard any really great results from any of these things. I mean, people keep binge eating and going to Weight Watchers and
Right.
Overeaters Anonymous. So do you think this might help people to eat less or binge eat less?
I think that it might. That said, one caveat is Dorsia who is the company that makes daridorexant, which is Quivic, tried clinical trials for that orexin antagonist in binge eating disorder and didn't find a signal. Animal studies do find a signal, and the one issue might be being able to control the behavioral phenotype that you're testing in animals compared to people. For example, it may work in animals, only in animals that are obese and have binge eating disorder, whereas in people, if you don't select for the combination of those things, you may not pick up a signal. So I think, it requires more study to identify the population that will be most responsive. There's a lot there that we don't quite understand, of course, in terms of mechanisms of actions of this system. But there's quite a lot of animal studies that indicate it can be effective in BED, binge eating disorder models.
Okay, yeah, that sounds great. Now, another group of people that are not, and they sometimes get confused with being addiction patients, and they're not, are people that are tapering off of, mainly benzodiazepines and then also other psych drugs. But the people with protracted withdrawal are taking, they're often misguided and doctors mistreat them. They taper them far too quickly, and then they have a longer period of withdrawal, or they taper very gradually, and there's just no good solution for them. They tend to be very sensitive to a lot of different medications, and they don't want to take more medication. But are you aware of it, that this in any way might help someone, maybe with the withdrawal symptoms? I mean, maybe even with the sleep issue, because they can't take, like you said, the Z drugs or the benzodiazepines. Those are things off limits to them. And
Right.
maybe one of these drugs would help with their sleeping or insomnia, and maybe with additionally withdrawal symptoms.
That's a very good point. I'm glad you brought that up because insomnia is a major comorbidity in all types of addiction. And the fact that these drugs treat insomnia well, I think, is important in their ability to also treat addiction in those subjects. We have what we call the dual hit hypothesis. So we propose that the orexin antagonists, like these drugs, work in two ways. One is you might think of bottom up, decreasing motivational drive for the drug in response to a stimulus that predicts drug availability. But the other way is by facilitating sleep. And you might think of that as having a top-down effect where that improves cognitive abilities. It improves executive function. And by improving executive function, that's going to facilitate abstinence because that will make it less likely that the subject would continue taking drugs in the face of predicted negative consequences, right? You could sort of think about it more and be able to control your behavior better when you have better executive function. So I think it can work in both ways, and the sleep effect could be an important element.
Yeah, yeah, that makes sense. I mean, just being able to get to sleep. I mean, that's people in early recovery, sometimes they might go to their treatment program, they might go to meetings and they get home and they're kind of alone with themselves and they can't fall asleep. And
Right.
and that's a dangerous time. So,
It is. Yeah, exactly right. Exactly so. And I agree that that is one avenue that these drugs also can be beneficial.
okay. Now, when you mentioned narcolepsy, that right away I was thinking of there's that other drug that does the opposite, I guess. It helps people wake up,
Yep.
modafinil. Is
Yep.
there some relationship to how those two different drugs work? One wakes you up and one helps you fall asleep?
Well, modafinil does stimulate orexin neurons. So the neurons that make this peptide, orexin, are responsive to modafinil. It activates them, and that might be one of the ways that modafinil drives waking. Um, in narcolepsy, what's interesting, though, is that narcoleptics, by and large, lack orexin neurons. So when studies were done initially in animals, people found that when they knocked out or eliminated the gene that makes orexin or the receptor for orexin, the animals exhibited narcoleptic-like symptoms, and that prompted people to look in the brains of human narcoleptic patients. And they discovered that they have almost no orexin neurons. And it seems like there's an autoimmune disorder in narcolepsy, often narcolepsy cataplexy, that kills off the orexin neurons. And so the idea is you're killing off these waking neurons, and that then drives sleep at inappropriate times, like you see in narcolepsy, cataplexy. So the two could be related. Modafinil might work by activating the remaining neurons that haven't been killed off by this autoimmune disorder. But that's one of the lines of evidence that have strongly implicated orexin in sleep and wake control is this lack of these neurons in narcolepsy, cataplexy patients.
Okay. And are you current like is this what you're like are you working on specifically a research study right now with this? Can you tell us
Yeah,
more about the specifics of that?
yeah, no, I'd be happy to. So we do animal studies, and we train animals, for example, to self-administer fentanyl or cocaine. And what we have found recently is when animals go through a paradigm that drives it, that makes them very strongly addicted to the drug, that actually increases the number of orexin neurons in their brains. So they have about 25 to 50% more orexin neurons when they go through a paradigm that makes them addicted. And it can be coke, it can be fentanyl, it can be oxycodone. All of these compounds or alcohol increase the number of orexin cells, which fits with this idea that the system is driving this hyper motivated response. We recently published a paper that proposes that what's happening is the orexin system normally can increase and decrease its expression adaptively. For example, there's more expression in daytime and less expression of this peptide in nighttime. And that's one of the ways it regulates its signaling. But what happens with prolonged drug exposure and addiction is it gets stuck in the upstate. So there's this what we call reserve population of orexin cells that turns on and doesn't turn off like it normally would, but it stays on in a persistent fashion. So that it is that the system overall is larger in a sense when it responds to conditioned stimuli that predict drug availability and drives a bigger motivational response than normal to salient stimuli. And these antagonists that are that important to sort of take that down a notch so that you lessen that hypermotivational response. But interestingly, you leave normal motivational activities in place. So that's important, right? I mean, you don't want to decrease all motivation for everything. You can't keep people from eating and having sex and other kinds of drives. Seems like this orexin system really becomes aberrantly engaged with hypermotivation, and you can use these drugs to take the edge off, if you will, or decrease that hypermotivation, but still leave baseline motivational processes intact.
Okay. So if someone were to take one of these drugs, it probably is not going to take away all cravings and drive to use the
No,
drug, but maybe just enough to make it tolerable early on.
exactly. To help them, you could think of it almost like water wings when you're learning to swim, right? I mean, it's going to help them navigate the initial stages, especially of abstinence, and maybe even long term help them remain abstinent by decreasing that hypermotivation that normally occurs when they encounter stimuli that previously have predicted drug availability. That's what happens with addicts. They will be in the environment, they'll be abstinent, they'll run into somebody or some situation where they previously took drugs, and that drives this craving response, in part because I think it's activating this orexin system. And so by decreasing that larger drive than normal, I think they're able to better navigate and remain abstinent.
Okay. Yeah, I just did just had an interesting interview with someone that she's an aromatherapist, and
Uh-huh.
as she was describing how that worked, I was thinking that, this is definitely something that can't hurt at all. I mean, why not? If it helps, you might as well offer it to anybody that has access to it. Um, and this isn't exactly the same because this is a real drug that has real side effects, but it does seem to be almost in that category of why not try it? It's not the same as Suboxone, where a person, you tell someone, we're going to keep you on another opioid for a year or more and see how that goes. And that's a big decision because they may want to try abstinence or some other kind of treatment. But this seems like a good adjunct therapy to other kinds of therapy.
Absolutely agree. Suboxone doesn't help the insomnia, right?
Yeah.
For example, and these drugs would. And so, yeah, I think it is these drugs are well suited for adjunct therapy, perhaps even for solo therapy in treating at least some types of addiction.
Yeah. Yeah. And yeah, Suboxone actually keeps people awake. I have to
Right.
tell people don't take it too close to bedtime.
Right. Exactly. Yeah.
So, yeah. So, um, what kind of animals do you use in the studies? Are they rats?
Oh, yeah. We use rats, and they have catheters that we implant in their jugular vein, and they hit a little lever and they get a shot of coke or fentanyl. They quickly learn to hit the lever, and that gives them IV drug, which they like and will work hard to obtain. We also give them light tones conditioned stimulus that's associated with those drugs, and those light tone stimuli by themselves will engage motivation that makes the animal work to find the drug. So we can measure drug seeking responses in this paradigm in response to those conditioned stimuli, and that way kind of mimicking what happens in the human addiction circumstance. And then we can manipulate systems in the brain. For example, if we decrease expression of orexin in animals, that decreases motivation as measured in our behavioral economics paradigm, very similar to giving them an antagonist like we're now talking about. We can measure activity in these cells and see that they're increased in association with the increased motivation for drugs. And we can see in animals that this increased expression of orexin is very persistent. So a rat will live about 2 years, 24 months. And if we make animals addicted and then just leave them alone, 5 months later, they still have this elevated number of orexin neurons, and they still exhibit this high motivation for the drugs that they haven't experienced in 5 months. So again, that captures, I think, an important element in the addiction area, where subjects are, in a way, once you're addicted, you're always addicted. That sort of rule, and I think that's in some sense true, and maybe in part because of this sustained change in the neurobiology of these orexin neurons. But now we have tools that allow us to manipulate and modulate, if you will, signaling in that system.
Yeah. Yeah, it seems like addiction is a really complicated thing. There's a lot of different things involved. You're focusing on orexin, and there's a lot of talk about endorphins and opioid receptors, and there's other I guess these different tracks in the brain, or different areas of the brain. What is it? The anterior tegmental system and the nucleus accumbens. Are those areas like related to this issue of the orexin neurons?
Yeah, I think they are. The ventral tegmental area is a major dopamine signaling system in the brain, and dopamine has been found to be a sort of common denominator among all kinds of motivated behavior. Orexin has been found to strongly modulate activity in those dopamine neurons in the ventral tegmental area, and we think that one of the ways it works is by modulating those cells. So when orexin is present, it increases the response of dopamine cells to the conditioned stimuli that drives motivated responding. By tamping down the orexin input, we can decrease that hypermotivation, but leave the baseline motivation more intact, because those cells are driven other ways as well as being modulated by the orexin system.
Okay, that's really interesting. I've another question which might go completely outside of the orexin issue, but there's in the use of opioid blockers like naltrexone long term, people are concerned. In the alcohol community, there's a method. What do they call it? The pharmacological extinction. Where they take naltrexone tablets when they drink, and then over time the drive to consume alcohol goes down over time, and they drink less and less.
Right.
And then there's people that follow that. They say, well, we don't want to block our endorphin system all the time. We want to reinforce good behavior, but not reinforce bad behavior. And I was wondering if is there any possibility that maybe that whole reward system is kind of like, for humans at least, a vestigial thing, that maybe we don't even need it, because there's people that do take naltrexone for years and years and even have implants and have these receptors blocked, and they seem to function fine. I mean, do we really need to have endorphins to tell us that's a good berry to eat and that's a bad one? Or is that more of like a caveman thing that we don't need that anymore?
Well, that's an interesting question. I think, like most parts of the brain and most systems in the brain, there's more we don't know than we do know.
Yeah.
And a lot of systems like the endorphins are modulatory systems. They're not, driving a direct behavioral response. So you can take them away, and on the surface, it can look like they're not having a major, deleterious effect. But, if you take it away long term, there might be effects that we haven't studied yet. There might be effects, for example, on learning, on memory, other cognitive abilities like that, which, motivation and reward are very important for learning and memory. You learn and remember things that are associated with reward. And so, one wants to be careful about long-term manipulations of systems in the brain that we don't understand all the aspects of. The naloxone treatment and that sort of pharmacological extinction study that you're talking about is interesting. One of the issues with extinction, though, is when subjects learn extinction, it's learning a new relationship. It's not forgetting the old relationship, right? So what they learn is when I take this drug, I no longer get the effect from it. But that has very little effect on some stimulus that was previously associated with them drinking, like going into a bar, and that stimulus driving a craving response to drink. So, it's a little tricky that way, and a lot of times those kinds of therapies are difficult to really implement in people in real life situations, and longer term, there could be consequences that we're not fully aware of yet.
Yeah, yeah, that makes sense. So it might be maybe a good idea, like when they say let's not take the drug every single day, only when we need it.
Right. Yeah. No, I
Okay.
think that's um, again, that would be something clinical trials would be really informative about.
Yeah.
For example, these orexin antagonists. First, let's identify subjects that they're likely to work well in. May not work for every case. And second, let's examine when it makes sense to take the drugs. Maybe not every day, maybe not in every circumstance, but maybe in social situations where there's a chance you might be exposed to stimuli that normally would drive a relapse response. That's possible that we can kind of tailor treatments around things like that after the initial period where you're helping drive the initial abstinence. Then there might be more targeted therapeutic approaches like that.
Yeah. Um, and now, what do you have you studied or worked with these issues of protracted withdrawal, like people who I talked about the people that consider themselves iatrogenically injured or injured by taking benzodiazepines, and they feel that there's some kind of a sometimes they call it toxic encephalopathy, that their brain has on some microscopic level been damaged, damage to receptors or changes, long-term changes in the brain. Um, does that sound reasonable that they've actually been, not that there's some big area of brain damage you can see on an MRI, but maybe something more subtle?
Yeah, no, I think that's certainly possible in the sense that even in our animal studies, like I was describing, we see very long-term effects of um, paradigms that make animals strongly addicted to drugs. And some of those long-term, if we leave animals alone for 5 months, they show a very strong uh, drive still for the drug. And we still see this elevated number of orexin neurons in the brain. Probably many other parts of the brain have persistent changes in gene expression as well that persist, that if we knew more about, we could start to think about therapeutics that would deal with those changes that have occurred that might be relatively permanent changes. It raises a whole nother question. If these are long-term changes in brain physiology and function, what are the mechanisms underlying those long-term changes, and how do we undo those long-term changes, right? Be it
Yeah.
an epigenetic change and that's very nascent research that we are really very new in the field, trying to understand the mechanism behind long-term changes in response to, for example, drugs of abuse. But certainly that happens, and whether it's damage, I don't know that I would think of it necessarily in terms of damage, but you can think of it in terms of a change that's occurred in the brain, probably in some kind of maladaptive way in response to prolonged exposure to these highly salient rewards.
Yeah. I had once heard, I don't know where I heard this, something about I guess from long-term stimulant use, that the little sacs on a neuron that hold the neurotransmitters, that they can become leaky. Is that a real thing that happens, or is that, maybe I'm not describing it correctly?
Well, you're probably thinking of vesicles that hold neurotransmitters in synaptic boutons, and the degree to which they operate properly. Stimulants do cause release of dopamine from those vesicles, and prolonged stimulant administration does have effects on the ability of those vesicles to hold the dopamine and to reuptake The dopamine, for example, Rodrigo, Andrade, and others have looked at those kinds of effects in animal studies. So I think it could be a real thing. I don't know that that's been established in people yet, but there are animal data consistent with that. And that might be another example of these sort of changes in brain that occur. How persistent they are, how easily they are undone, or how they could be treated pharmacologically, is still an open question and an area of active research.
Okay. And I think one good thing, even if people listening to us are concerned about long-term brain damage, the good news is I think that brains are good at compensating and adjusting to changes and damage.
Absolutely right. Absolutely right. Brains make new neurons all through adulthood in particular parts of the brain. Even aside from making new neurons, for example, these orexin cells can increase their expression of that neurotransmitter molecule and decrease that expression. They kind of wax and wane, usually adaptively. Something happens with prolonged drug exposure that gets them stuck in this upstate, and the reserve orexin neurons continue making a lot of orexin. Um, so it's not damage, it's a change. And knowing what that change is then opens the possibility for being able to reverse that change, as well as to give an antagonist that can block the effect of that elevated orexin signaling.
Okay, great. And another thing I was just curious, and this may be related or not, is that I had a discussion recently with someone that he, provides ketamine therapy, and he was talking about stimulating the growth of dendrites, and I don't know how significant that is. Is that are dendrites, are those the things that connect one neuron to many neurons? Like one neuron can connect to like maybe thousands of other neurons?
Yeah, dendrites are the major receptive surface of neurons to receive input from other neurons. So they're called dendritic trees. Dendrite is tree, right?
Yeah.
The word, it means tree. And so there are highly branched structures that emanate from a cell body of a neuron, and they receive lots and lots and lots of inputs, and they receive inputs from many different structures that way. And so increasing the number of dendrites increases the opportunity for those cells to be modulated more or less by the inputs from different areas. There are data in animal studies, again, that show in the nucleus accumbens that there are changes in dendrites during drug exposure that increases their receptivity to dopamine inputs from the VTA. And that's one of the ways that the hypermotivated responding occurs to drugs. But in turn, that dopamine input in the VTA, the ventral tegmental area, is regulated by orexin. So by blocking that orexin input, which increases activity in those cells, we can decrease signaling in that whole pathway, including the increased dendrites in the nucleus accumbens.
Okay, so just the act of growing more dendrites is not necessarily a positive thing. That could work against us if they're, getting more of them in the wrong place.
If they're driving maladaptive behaviors, right? If they're
Yeah.
underlying the ability of cues associated with drugs to drive craving for the drug, then that's not a good thing.
Okay, that would imply, I think, that therapy is very important, or at least a person, being aware and making positive changes in their life, psychological things rather than just worrying about the physical neuroanatomy.
Absolutely agree. And I think that touches again on this top-down kind of element in the addiction process, where increasing executive function increases top-down control over behavior, and awareness of your problem and awareness of the negative consequences of your behaviors, I think, can help you modulate your behavior and remain abstinent in a longer term. Increasing sleep is one way to increase that executive function, but therapy, right, is another way that I think one could increase executive function in a beneficial way. Maybe a combination of orexin antagonists with therapy could prove to be quite a strong approach.
Okay. Do you of the three brands currently available, of those sleep aid drugs for insomnia. If just say that, a doctor has a patient who asks for, just me, for example, say later today, a patient says, "Do you recommend something good for sleep? Could you give me a prescription for something?" Do you have any reason to prefer one over the other?
It's very early days. They're all pretty recently um, approved drugs and have been relatively little studied in the addiction paradigm. Balsamra has probably been studied a little more in the addiction paradigm just because it was the first that was approved.
Yeah.
But there's no reason to think it would be better than Dayvigo or Quivik at
Yeah.
this.
Yeah. When were I think you had said it earlier, when was Balsamra approved?
I believe it was 2014.
Oh, okay. Yeah, because it
Right.
somehow that name stands out more to me that, I feel like there may have been like a company drug rep that came in and talked about it at some point.
Yeah, that could be. It's made by Merck. Um, and I believe it was approved in 2014, and they've been marketing it recently on TV. You'll see Quivik. That just recently showed up in commercials on television. I've seen it recently to treat insomnia. And that's made by Idorsia, which is a Swiss company. It's quite a good drug, I think. They have evidence that it has some advantages over Valsamra, but every company has evidence that their drug has advantages over another company's drugs. I think they're all probably good drugs, and it may depend on the patient. And the biomarkers, the kind of things that are driving them to an addiction profile, might favor one over the other for a particular patient, but we don't have the studies yet to really guide us well enough, I think, to choose among them at this point.
Okay. And so are you at Rutgers in New Jersey?
Currently, right now, I'm not, no, but
Okay.
that's where my position is. That's where I direct the Brain Health Institute, and that's where my lab is, where my day job is,
Oh,
where we're
okay.
experiments in animals and doing these self-administration and addiction studies.
Okay. Yeah, it seems like it might just be there's that really popular podcast by another brain scientist out of Stanford. So I don't know. It seems like they must be doing a lot of brain research there also, or at least you hear about it a lot.
Oh, yeah, well, Stanford's a very strong place in many sciences, including neuroscience. I'm not familiar with that podcast. I missed that one, but
Oh, it's Andrew Huberman. He's really good. He's yeah, you might want to look him up. He's very easy to understand. So like the public loves him, but he's a also a neuroscientist.
I'll look into him. Andrew Huberman. Yeah. All right.
So
Thanks for the tip.
do you have you done any podcasting like on your
It,
own?
I have not.
Yeah, it's fun. So
Yeah,
you
no, it is. No, this is a very interesting, enjoyable conversation. Now,
yeah.
you're a great guide to it.
Oh, thank you. Yeah, it's yeah, I love doing it. I love podcasting. I don't really could never be a broadcaster. I don't, but yeah, I love asking questions, having conversations.
Yeah, well, you
So
ask a lot of good questions, and I think by asking questions and getting answers this way, it's very informative to people. It's like a Q&A session after a scientific talk, right? You get a lot of information out of that, a lot of times more than you did from the presentation itself.
oh, definitely. Do you teach undergraduate students at all?
I don't teach undergraduate students right now. I used to earlier in my career,
Yeah.
but I've become more kind of administratively oriented and direct the Brain Health Institute at Rutgers that includes over 300 neuroscience labs,
Oh,
uh,
wow.
900 trainees. So that keeps us busy when we're not in the lab doing experiments.
So what are some other like interesting, notable experiments going on like outside of like in your lab under your direction?
Well, the other major um, set of experiments that we're doing involves another system in the brain called the locus ceruleus, which is a major norepinephrine system in the brain. Also is involved in regulating sleep and waking. But our prior studies with that system show a role in attention and behavioral flexibility. So more executive function, cognitive sorts of processes. Um, one and it is implicated in many different disorders, including depression, sleep disorders, circadian disorders, and memory problems. It, for example, is one of the earliest pieces of the brain that degenerate during Alzheimer's disease. In fact, it's the earliest known place that starts to show damage during Alzheimer's disease. So one would like to be able to manipulate these cells. The problem is they're located right in the middle of your head, in between your ears. And so it would be quite invasive to go in and stimulate these neurons. We found recently a circuit that goes from the suprachiasmatic nucleus, which is an area in the brain that controls circadian rhythms, to the locus coeruleus. Indirect circuit goes through another part of the hypothalamus. The interesting thing about the suprachiasmatic is it gets its inputs mainly from the retina. And so that then opens up the possibility of regulating this locus coeruleus from the eye. And so we recently developed a method for doing that. We use what's called chemogenetics, which is a way to insert a gene into retinal ganglion cells that then allows us with eye drops to stimulate those retinal ganglion cells, which in turn project to the suprachiasmatic and activate the locus coeruleus. And in our animal studies, that is effective in preventing or even reversing depressive-like behavior in response to different experiments. For example, light deprivation causes a depressive-like syndrome or stress. And also, we recently have shown it's effective in improving memory and behavioral flexibility in a genetic Alzheimer's model in rats. So we're very excited in driving that forward as a potential therapeutic in people. That approach, I think, is quite feasible in people. We have a patent on that and kind of working with venture capitalists and biotech companies to try to develop it as a therapeutic for treating a variety of neuropsychiatric disorders. So that's the sort of other major project in my lab.
Oh, that's incredible. And I know we definitely need more treatments for Alzheimer's. I remember when I practiced in a much busier family medicine practice, it was terrible to see people decline with Alzheimer's and very quickly, and it became a very difficult burden for the family members to take care of them.
Absolutely.
And and then when the drug companies would come in and tell us about these different drugs, that none of them really, not only did they not reverse the symptoms, they didn't even stop them. They slowed the decline, the rate of decline. It almost seemed like torture. It's like you're going to go through this year of torture, but maybe we can stretch it out a little bit longer. And it became this moral dilemma, like, do we give these drugs or not? Because they're not really going to do much to help. But yeah, it would be great if there was something much more effective.
Yeah. Well, it's early days. Again, these are only animal studies so far, but we see pronounced improvements in memory and behavioral flexibility in our genetic model of Alzheimer's. It's a treatment, not a cure, though.
Yeah.
So, it's not preventing the damage that occurs, but it's able to stimulate the remaining locus coeruleus neurons and compensate for the damage in locus coeruleus that happens in Alzheimer's disease. And I think, it could be a longer term treatment that might be effective over, years of therapy, we're hoping, but we need to do more
Yeah.
studies to really map that out well.
Do you work with I was thinking about this because somebody asked me to write an article about it, and it was interesting. Uh, Transcranial magnetic stimulation, is that useful to you, or is that specific enough for your use?
We don't work with that. Typically that is in either monkeys or people.
Okay.
There's a little bit of TMS that's done in rat, but it's so available for study in people that, a lot of times they just directly study it right in people, sometimes in monkeys.
Yeah.
We haven't done any of that, but it's been found to be somewhat effective for some disorders in people. Um,
Yeah.
some evidence for effectiveness in depression, as I remember. That's the main one that I remember so far with
Yeah,
TMS.
apparently there's like a deeper version, I guess like the the deep one can get as deep as like 4 or 5 centimeters or something, and then that one, the deep TMS can work on OCD also,
Right,
which
right.
I
Now,
just learned this a couple of days ago.
yeah. Yeah, no, these techniques are advancing quickly, all the time with new methods to affect more areas in the brain and be able to triangulate and target deep brain structures, not just the surface structures. And so as they're able to do that, they'll be able to, treat more disorders. That was part of the thing that we're getting at with our retinal stimulation, is being able to get to this deep brain system, which is a fundamental system for cognitive processes like attention and behavioral flexibility. Otherwise, is difficult to target.
Yeah, yeah, that's incredible. So you're actually changing a gene in the eye or something, and
Yep.
creating a new photoreceptor somehow that goes,
Well,
or is
actually,
it?
we insert a new gene into the eye that itself doesn't do anything, but it's a receptor for a particular compound that we can give, which itself is inert in the body. So it's a sort of lock and key arrangement
Oh,
where you
wow.
cause this gene to be expressed. It just sits there, doesn't do anything. But you can give this drug, in our case, is an eye drop, which only affects those receptors. It doesn't do anything else. So
Oh,
it's a
wow.
very selective, targeted, kind of targeted gene therapy approach, but different from other gene therapy approaches because you're not affecting genes throughout the body. You're only targeting these custom genes that you've engineered. These are engineered genes that you've caused to express in the eye, and then you target them with this engineered drug that selectively acts on those receptors.
Yeah, that's incredible. So there's definitely things to look forward to in the future. There's definitely
Absolutely.
things in the works, in the pipeline.
Absolutely. That chemogenetic approach, even not necessarily in the eye, applied to other parts of the brain, I think will also turn out to be a very useful approach for neuropsychiatric disorders.
Okay. Thank you. Dr. Gary Aston Jones, I want to thank you again. Thank you for joining me today on the podcast.
Sure, Mark, it's been great speaking with you. Thank you for having me.
Thank you.